This abstract is about functional MRS at 7T to measure changes in GABA and glutamate within the visual cortex due to peripheral and central visual stimulation. The blood oxygen level dependent signal increases during active stimulation of the central visual field, and this is reflected in the fovea of the eye. However, in the peripheral stimulation, the fovea is no longer directly stimulated, and there is a negative bold signal. The positive and negative bold activation is hypothesized to induce an imbalance in the excitatory and inhibitory neurotransmitters within the primary visual cortex, designated by V1, corresponding to the fovea of the eye. GABA is hypothesized to increase in V1 with peripheral visual stimulation, meaning the fovea is not directly stimulated. However, glutamate, which is an excitatory neurotransmitter, this neurotransmitter is hypothesized to increase in the V1 with central visual stimulation. These changes in these metabolites can be measured with functional MRS. We apply this at 7T because glutamate is more easily detectable at this high field strength. The difference in GABA and glutamate can be induced by different paradigms or functional tasks. The goal here is to measure differences in the levels of GABA and glutamate in the V1 region of the visual cortex due to central and peripheral visual stimulation, and to determine a test and retest reliability of these changes due to the peripheral stimulation. A non-edited semi-laser sequence was used to measure spectra within the visual cortex region, designated by the yellow box as seen in part B of this figure. And this is the V1 volume of the visual cortex. The voxel had a volume of approximately 3 ml within this region. Spectra were measured under conditions of both central and peripheral visual stimulation. Each dataset consisted of 3 runs or experiments, 2 with peripheral stimulation and the other with central visual stimulation. Each of these runs or experiments had 12 alternating blocks of active or inactive stimulation. The TE that was used for these experiments was long at 80 milliseconds or short at 29 milliseconds. And the quantitation of the metabolites of interest, GABA and glutamate, was done with LC model. We also measured percent differences in GABA, glutamate, glutamine, and the combination GLX, glutamine plus glutamate in both between the off and on active states of stimulation. A test-retest reliability was also evaluated for 2 runs of peripheral visual stimulation, and this reliability was measured in terms of the intra-class correlation coefficient, or ICC, for GABA and glutamate concentrations. This is the experimental setup describing the run and block patterns of the stimulation. On the bottom left, you have the visual stimulation, central visual stimulation, which is on. And in the middle, you have peripheral visual stimulation on. And in both cases, when the stimulus is off, only the central dot is displayed. And the task of the participant is to be able to record what the color of the central dot is as the experiment is running. Each block consisted of 48 seconds. This is some of the resulting spectra that was measured in the yellow region of the visual cortex due to stimulation of the fovea, direct stimulation of the fovea due to central visual stimulation. The blue spectra is in the off state, and the red spectra is in the active state. There are minute differences in the spectrum which reflect the different levels of glutamate and GABA, as well as glutamine. Quite simply, there was a small trend of increase in GABA from the on versus off stimulus states. However, the trend in glutamate was more inconsistent. But the combination of glutamine and glutamate showed a more predictable trend, a decreasing trend. In terms of the test and retest reliability, GABA showed a much more high correlation between the two peripheral runs across the 4 volunteers. However, glutamate had a much lower level of correlation in terms of the interclass correlation coefficient. Again, GABA showed an increasing trend of 3 to 10% increase of concentrations in the peripheral visual stimulation, which is correlated with its inhibitory nature. With central stimulation, it decreased, again, due to its inhibitory role. However, glutamate did not show consistent trend, either decreasing or increasing with any type of peripheral or central stimulation. However, the combination GLX was stable in both the central and peripheral visual stimulation. Again, the retest reliability for GABA is higher than the levels for glutamate in terms of the ICC. More optimization of the sequence is needed to be able to more reliably detect these and measure these changes in both GABA and glutamate. Overall, the increasing or decreasing trends in GABA and glutamate are not statistically significant. However, the hypothesis remains to be convincingly shown that negative and bold positive signals are correlated with an imbalance, a transitory imbalance between the inhibitory and excitatory neurotransmitters, particularly GABA and glutamate, which are better detectable at 7T. However, in this study, the experimental paradigm as well as the choice of pulse sequence, including whether to use long or short TE, needs to be further evaluated and refined in order to be able to more accurately detect these changes. Acknowledgements are given to the USC Laboratory of Functional MRI Technology and the Laboratory of Neuroimaging. Thank you.